John, I don't know if you're originally from Seattle, but did his undergrad at the University of Washington, and then moved to the East Coast to Yale to join Joe Croft's lab, where he studied T cell biology and the role of transcription factors in regulating T cell biology, presumably mostly in animal models. And then moved over to the Broad Institute, MIT and Harvard, to Neer Hokkanen's lab, where he started working on trying to understand how genetic variants, autoimmune-associated genetic variants, impact cells, which I think we felt as the group that invites people to give talks mapped with a lot of what we're trying to do at Seattle Children's in CIIT. So we thought he'd be a great speaker to have on, and welcome back to the Pacific Northwest.
Thank you, Sean. I really appreciate being asked to come speak. And I just started my lab at BRI a year ago. And we're continuing to work on this problem where we're trying to understand genetic susceptibility to autoimmune disease. And obviously at BRI, autoimmune disease is a big interest. And in my lab in particular, we're interested in the genetics of disease. And the reason that is because there's a large heritability of most autoimmune diseases. So here we're depicting diabetes. Can you see my cursor? Or, okay.
Yes, we can.
Let's assume you can. Okay, sounds good. So here we're depicting a monozygotic twin study where the coincidence of both twins developing diabetes is around 65%. That suggests that there's a really large genetic component of disease, but it's actually quite complex, just like the disease itself. So in order to really nail down the regions of the genome that are associated with disease, we use genome-wide association studies, which are when we take large numbers of cases and large numbers of controls and we genotype them, and then we can look to see which genetic variants are preferentially happening in disease versus not in disease. So then we can create these Manhattan plots where the y-axis here is the association p-value, and the x-axis are all the different chromosomes in the genome in different colors. And then all these little dots are genetic variants. And if they're above this genome-wide significance line here, then they're considered significantly associated with disease. So if we zoom in on one of these regions, we can see that there's not just one genetic variant that seems to be associated in this region of the genome, but there's many. And this R-squared metric is a measure of correlation between genetic variants. So you can see that there's many tightly correlated variants in this region that are highly associated with disease. Most of these variants are non-coding, and there are many different genes in this region. And it's also possible for these variants to regulate genes that are not in this region. Given that non-coding regulatory biology is really complex. So, so the reason we sort of see this pattern in lots of tightly correlated genetic variants in one region is because of linkage disequilibrium, which is the non-random inheritance of genetic variants in blocks. And the reason that is because recombination is not random in a given population. So we tend to inherit genetic variants in blocks. So the causal variant for a specific disease may be inherited along with a bunch of other variants that are not doing anything with regard to disease. So then when we go back and look at this plot, we see that this could easily be explained by one or a small number of genetic variants that are causing disease, and then the rest are just kind of along for the ride. So a big problem in post-GWAS is trying to understand which genetic variants are driving disease, and this could actually help us to unveil some really important disease mechanisms that could help us, better treat autoimmune disease down the line. And so if we could pinpoint these causal genetic variants away from all of these other like passenger variants, we could understand better what the mechanism is on acting on specific disease cell types, and what physiological processes that are disrupted downstream of that, and how that eventually leads to disease. And that's really the goal of the work that we do in my lab. So I mentioned most of these genetic variants are in non-coding regions, and inside of those regions, they're enriched in immune enhancers. So an enhancer is a region of the genome that's non-coding, and it's a region that basically associates with promoters of genes and helps to drive their expression. So that's why it's called an enhancer. It enhances expression of a target gene. And so genetic variants in these enhancer regions could block transcription factor binding, and transcription factor binding is required for this enhancer region to interact with this promoter. So if a genetic variant disrupts this transcription factor from binding, you could imagine how that might disrupt downstream gene expression. And we can read out these putative regulatory regions in the genome, these putative enhancers, using techniques that essentially look for regions of chromatin that are more accessible, because that's where transcription factors bind in the genome. It's essentially a region that needs to be accessible for that transcription factor to bind. And so we can use techniques like ATAC-seq that insert transposase into preferentially accessible regions that are nucleosome-free, and then it inserts these, it fragments the genome and inserts these primers so that we can amplify those regions that are preferentially accessible. And then what we get from this, after we amplify those regions, are these peaks in the genome. So this really tells us that this region is more accessible and nucleosome-free and a putative enhancer. And then we can take all the disease GWAS that we're interested in and see if the SNPs are enriched in these regions of accessible chromatin. And this is just a picture to show you how cell type specific these regions of accessible chromatin can be. This is DNase hypersensitivity. It's another method for accessible chromatin. And this is from John Stam's lab at UW, a recent paper. And so you can see that while there could be constitutive regulatory elements that are just accessible in every cell type, there are many elements that are actually very cell type specific. And so we can use this as sort of a readout of cell type specific regulatory regions. So in my previous work, I took a bunch of this accessible chromatin data from many, many different cell types and took these autoimmune disease GWASs, so all the SNPs from these GWASs, and basically asked whether there are specific cell types where these genetic variants fall within accessible chromatin. And so you can see for many different autoimmune diseases, we get specific enrichments in T cells, but also in B cells. I know there's a big interest in B cells here. And so, but what I'm going to be talking to you today about is trying to target these causal variants in T cells and how to follow up on variants. So really what I'm going to be going through today is techniques for identifying disease causal variants and how do we define their mechanism. And then once we sort of understand their mechanism, how do we know what they're doing on disease relevant cell types to then lead to disease? So in my postdoc, I did a lot of work using many different techniques to understand which technique is actually best at identifying disease-associated variants versus just other non-disease associated variants. So we used methods that involved looking at the chromatin contacts. So part of what I showed you earlier, an enhancer is a region that contacts the promoter. Well, we can take that information through performing something like 4C, high C, or high CHIP, which are all taking into account chromatin contacts. And I've already explained ATAC-seq. So we can look to see whether variants that are in regions that contact other promoter regions are enriched for disease-associated variants versus non-disease-associated variants. And we can do the same for ATAC-seq as well. We can test putative enhancer regions or just regions containing a genetic variant for whether that region regulates gene expression through targeting CRISPR, versions of CRISPR to that region. So if we target CRISPR-I, which is CRISPR interference, which basically shuts down regulatory DNA activity. So if we target that to a variant and find that it reduces the expression of a target gene, then that's a good indication that it's in an enhancer region that's important for that gene activity. Conversely, if we use CRISPR-A, which is a form of CRISPR activation, and if we target that to a variant, and it is putatively a regulatory region that drives expression of that gene, you may expect more expression of that gene. And then we also tested transcriptional reporter assays, which are essentially cloning the genetic variants with maybe 200 base pairs of surrounding sequence into these reporter plasmids. You could test the major allele versus the minor allele and see if there's differences in expression conferred by the major or minor allele. And then we can computationally predict transcription factor binding and also perform transcription factor ChIP-seq to see whether a given variant is predicted to be disrupting transcription factor binding. So we did this for 4,000 common variants in the T and FAIP3 locus. Around 300 of them are disease-associated. And I should say that we chose this locus because there's around 15 independent associations for various autoimmune diseases. There's tons of different disease associations in this locus, and we've done some haplotype analysis to determine that there was around 15 independent associations. So it was a very dynamic locus, and that's why we wanted to study it. So with all these techniques. So to summarize a very, very long project, we've basically assessed whether a given assay is likely to identify disease-associated variants versus non-disease-associated variants. And so to depict that, we have this precision recall curve, which is essentially a readout of sensitivity and specificity of the assay. So on the x-axis, recall really means sensitivity, so the ability for a specific assay to pick up all the disease-associated variants versus non-disease-associated variants, or regardless of whether they're picking up non-disease-associated variants. And then precision is specificity, so whether a given assay can pick up disease-associated variants and specifically not other common variants. So just from looking at this data here, we see that CRISPR-I is performing pretty well at identifying, with sensitivity and specificity, at identifying disease-associated variants. But what we know about these non-coding variants that are likely to be disrupting regulatory region activity is that there's likely needing to be an accessible chromatin to do so. So if we look only at variants that are inaccessible chromatin, we get a very different curve where we actually now see that transfection MPRA is performing pretty well, especially from a sensitivity standpoint. And that's not such a bad thing because then we can test more variants in this assay to see whether they're likely to be causing disease. And so really the combination of an MPRA, which I'm going to explain later, it's a form of a reporter assay, and CRISPR-I are the best methods at identifying disease causal variants that we've identified. And notably, CRISPR-A and transcription factor motif disruption are not good techniques, at least to start with, to try to identify disease-associated variants. So I talked a little bit about MPRA. I'm actually going to go into what MPRA is. it's kind of like a luciferase assay where we can clone in 200 base pairs of the surrounding the variant and then each variant allele into independent constructs, luciferase constructs. So here we have 200 base pairs of genomic context with the variant in the middle and a minimal promoter and then luciferase. And if we transfect cells with this construct, we can measure whether a specific allele of a variant is more likely to drive more expression than the other allele. And so we call this allele-specific expression. And it's sort of a readout of whether a variant is likely to disrupt transcription factor binding. So we can do this on a massively parallel scale by cloning in tens of thousands of variants. And instead of using luciferase, we can just use barcodes to read out the expression of each variant allele and then compare each variant allele to each other to see which variants are driving allele-specific expression. So we decided to do this on 5 different diseases, including type 1 diabetes, multiple sclerosis, rheumatoid arthritis, IBD, and psoriasis, totaling 580 areas of the genome that are associated with disease. And that's almost 20,000 variants that are associated with disease. And we chose to perform this assay in jerkats because they're a decent model of T cells, and we can get hundreds of millions of cells and be able to perform this assay with high fidelity. Just to give you a look at what the data part of our analysis pipeline looks like. So I told you that we barcode each element, so we call those barcodes tags. So each variant allele can have as few as 10 tags or as many as 10,000. So that's depicted here on the x-axis, and on the y-axis, we can determine whether a specific variant element is being expressed more than you would expect by chance. And so you can just take the RNA expression of that barcode and compare it to the plasmid prevalence of that barcode. And if it's more or less than you would expect by chance, then it's an active element. And we know this is performing well because we put in a bunch of negative controls that are not supposed to be driving expression of the reporter plasmid, and also a bunch of positive controls that do seem to, as a majority, drive expression in the assay. And in green here, we have our library of variants that we've tested in MPRA. And we believe this is doing a pretty good job, just because when we take these active elements and we look at where they are in the genome and specifically where they are in accessible chromatin and of various different cell types, we see that we're getting large enrichment in T cell and hematopoietic cell regions of accessible chromatin, and specifically not non-hematopoietic cell types like neurons. So we believe that we're picking up regions of accessible chromatin that are relevant to the cell type of choice. So really what we did the assay for is to find variants that drive differences in accessible chromatin. So on this plot, on the x-axis, we have the maximum expression of a given element. And so if an element is expressed more than by chance, like I showed you on that previous slide, it's called a putative cis-regulatory element. So that means that if it's driving more expression than by chance, it could be in the genome acting as a regulatory element. And on the y-axis here, we have allele-specific expression. So does one of the alleles actually cause more expression than the other allele? And those are depicted here in purple, and we call them expression modulating variants. So one way for us to essentially test whether our assay is picking up relevant information is through looking at whether a given variant is capable of disrupting transcription factor binding because that's a major mechanism that we believe that the assay is measuring. So we can compare the TF binding score allele difference. So based on whether an allele, if you disrupt that TF motif, does that drive a reduction in expression? Or conversely, if you're building a TF motif, does that increase expression? And so we get a really good correlation between the allelic bias in the assay and the predicted transcription factor binding scores. So that indicates that our assay is doing a good job of reading out transcription factor binding. But probably the best way to score a given assay that we've come up with is by using a totally orthogonal measurement, which is just statistical fine mapping. So statistical geneticists can actually perform fine mapping, which is basically assigning probabilities to variants in each GWAS locus. So some variants are much more likely to be driving disease than others. And so we can look at the probabilities of each of those variants for driving disease and see whether our assays are doing a good job of finding the variants that are more probable of driving disease versus the ones that are less probably driving disease. So here we have probability for causality, and 0.5 is 50% or above probability of driving disease. So here DNA sensitivity, which is just alleles, accessible chromatin readout, is increasing our ability to find causal variants in the genome about 12-fold. If we look at our MPRA assay, we're getting almost 30-fold increase in our ability to find causal variants in the genome. And if we combine both assays, we get almost 60-fold likelihood of identifying causal variants in the genome. So this really tells us that the MPRA does a really good job at identifying non-coding causal variants in the genome. And if we couple that with DNase hypersensitivity or other allele or other accessible chromatin readouts, that we can actually improve our odds of finding a causal variant substantially. So here I've mapped all of the variants that were in accessible chromatin that were hits in our MPRA assay. And here you can appreciate that the fold change brought about by each variant is very low, which is pretty much what you would expect from GWAS. So most odds ratios from GWAS are below 2. And so we actually find that the differences in expression conferred by each variant are pretty low. And so these are all the different diseases that we studied. And in purple, you'll find whether that variant is associated with that disease. And directionality, so whether risk allele drives more or less expression of the reporter. So you can appreciate that there's quite a lot of risk alleles that actually drive more expression of the reporter, which is really interesting. And down here, I've listed all the nearby genes that are either EQTLs, which are dark blue. So EQTL is whether that variant is associated with expression of that gene. So the risk allele may drive lower expression of the gene, for instance. And so we can see that there are many EQTLs for genes that are important in T cell biology or just in immune cell biology. And in gray here, they're just genes that are nearby. So we think we've picked up a lot of really relevant regions of the genome that are playing a role in T cell biology. So now that we've done our assay and we know that we've enriched quite a lot for causal variants in the genome, what can we do to better define their mechanism? Well, I've already talked to you about CRISPR perturbation methods, such as CRISPR-I and CRISPR-A. And while CRISPR-A isn't good at enriching for causal variants as a first pass, it could still be good at identifying target genes downstream. So we've previously utilized CRISPR-I in the TNFA-IP3 locus to identify cell type specific enhancer regions. So here I have a TAC-seq, which is accessible chromatin, which are these little peaks here on this track. So you can see that U937 cells are monocytes, GM12878s are B cell LCLs, and JERCATs are T cells. So these cell lines all have specific regions of accessible chromatin that seem to be designated by cell type, and some are common to all cell types. But you can appreciate how we can use this assay to really identify regulatory regions in a very cell type specific fashion. And so I'm going to show you a few examples where we've used CRISPR-A to try to identify the target gene of specific variants. So a very talented summer student, Victoria Lash, did some work in the lab to try to follow up some of these MPRA variants. So TA gap, here, this variant is about 50 kb upstream of TA gap. It's inaccessible chromatin, as you can see by this red dot here. And it also is one of the only variants in this locus that has allele-specific expression according to MPRA. And it's the only variant that has both. It's preferentially inaccessible chromatin in regulatory T cells. And there's some evidence for this regulatory region, putative regulatory region, to be connected to the TA gap promoter using promoter capture Hi-C. And so Victoria applied CRISPR-A to both the transcription start site of TA gap as well as to the site of the variant using CRISPR-A. And when we use guides that are targeted to the transcription start site, we get a noticeable increase in expression of TA gap. And if we target guides to the variant, in some cases we do drive an increase in expression, although it's much to a much lower extent than the TSS. So we think this is a hint that TA gap is the target gene of this variant, and we have a lot more work to do to follow that up. We've done the same thing with CD28, where we see a large increase in expression when we target guides to the CD28 TSS. But when we target guides to this variant, which is 60 kb away from the gene, we actually see a substantial increase in expression for various different guides. And we were particularly interested in this variant that's in the BOC2 region. So it's actually in the second intron of BOC2. And it's 30 kb away from the BOC2 TSS, which it looks like a small distance, but it's actually 30 kb. And there's some, it's in accessible chromatin, preferentially in naive T cells. And it contacts the BOC2 promoter specifically in naive T cells and not other cell types. And we were really interested in this variant because BOC2 is known to suppress effector T cell differentiation and instead promote regulatory T cell differentiation. So, this would make sense in the context of autoimmunity. If the variant actually reduces BOC2 expression, then you may expect there to be actually more effector T cells and less regulatory T cells. So in order to follow up on this variant, we used a base editing approach where we could precisely replace the non-risk allele with the risk allele using a cytosine deaminase conjugated to a Cas9 nickase. And from that, we can get a pool of edited cells and unedited cells. And what we can do with that pool is we can then sort on BOC2 expression. So we can sort on bins of low BOC2 expression and bins of high BOC2 expression and see which bins actually enrich most for edited cells. And what we found is in the context of the risk allele, there's actually a reduction in BOC2 expression. And so this is basically the model where we see in the context of the non-risk allele, there's enhancer activity that drives BOC2 expression. In the context of the risk allele, we have a reduced enhancer activity, and that leads to reduced BOC2 expression. So this variant is very highly specific to T cell accessible chromatin, as we don't see accessible chromatin overlapping the variant in B cells or in monocytes. It's in a very highly conserved region. It overlaps ETS1 binding region, according to ChIP-seq, and it's predicted to disrupt both STET and ETS1 binding motifs. And we believe that there is a difference in transcription factor binding, because if we look at heterozygotes that have one copy of the risk allele in their CD4 and CD8 T cells, and we perform a TAC-seq on those T cells, and we just look for the prevalence of the non-risk allele or the risk allele in those ATAC-seq reads, we actually see many more reads in the context of the non-risk allele than the risk allele. So this kind of gives us a hint that there could be differences in transcription factor binding conferred by the variant. And another hint that the variant is only acting in T cells is through EQTL studies. So EQTLs, again, are associations with genetic variants to expression of various genes. So you could basically do RNA-seq on hundreds of individuals on several of their cell types and see which genetic variants associate with the expression of each gene. And so in this case, we see that there's a reduction in expression conferred by the risk allele of BOC2, of this RS7292838. But this is only really seen in the context of naive T cells and not in activated or effector T cells or in other immune cell types. So this really tells us that the effect is pretty much happening in naive T cells. So we have a good idea of what the mechanism is for this variant. How can we understand better what's happening in T cells? Well, I'm a immunologist, and I worked mostly in mice in graduate school. So we looked at the possibility of creating a mouse for this variant. I mentioned that it was really highly conserved. So here I've plotted human chromosome 6 and mapped all the mouse chromosomes to it using a synteny analysis. And you can see that even down to the base pair, the variant is very highly conserved between human and mouse. And it's even distance-wise relatively similar to the human from the BOC2 TSS. And it's super highly conserved among many mammalian species, this whole motif. So it's probably very selected for. The region in mouse also binds STAT and ETS proteins, TFs, in various different T cell types. And it's predicted to disrupt both ETS and STAT binding motifs, so the mouse binding motifs. So a talented postdoc in Ryan Tui's lab in Jackson Labs created a mouse with a deletion of the variant, 18 base pairs, so that we could essentially test in vivo whether there was an effect of the region containing this risk allele on T cell biology. So just upon initial RNA sequencing, we found that there was, a difference in transcriptomes from mice that have this small deletion and between wild type littermates, and a slight, barely significant difference in BOC2 expression. And when we looked, we've done Westerns, and the protein looks normal in these animals. So in order to test whether these T cells are perturbed in some way, we chose to do a highly controlled model where we take naive BOC2 18-del cells that are crossed to the OT1 TCR transgenic and co-transfer them with wild-type OT1 cells into wild-type recipients, and then infect with VSV ova to track the T cell responses downstream. And a lot of this work was done by Michelle Lissner in Dan Campbell's lab and Ingrid in my lab. So you can see that upon transfer, there was a 50-50 ratio of cells. And then at 7 dpi, we actually see that the BOC2 18-del cells have a substantial reduction in proliferation or expansion compared to wild-type cells. And this is, we did the statistics on this here, and it's actually very reminiscent of when Rochaudhary et al. Did the same co-transfer experiment with BOC2 knockouts versus wild-type cells. So over time, they see quite a large reduction of BOC2 knockout cells in recipient animals. And so this is at day 7, we see this pretty measurable reduction in these cells. We think this is due to terminal effector differentiation, and I'll get to that in a moment. So Sue Keck wrote this review along when I was in grad school about how to identify terminal effector cells versus memory cells. Terminal effector cells are the cells that go out and basically kill virally infected cells in the body. And once they're terminally differentiated, they stop dividing. So they express KLRG1, but they no longer express the IL-7 receptor or CD62L, whereas less differentiated cells, such as memory cells, are KLRG1 low, but IL-7 receptor high and CD62L high. And notably, there's differences in transcription factor expression, where memory cells express a lot of EOMs, terminal effector cells express much more TBET. So we tested our mouse to see whether there was differences in the expression of these proteins. And so first we looked at memory versus effector, just looking at CD127 and KLRG1. And when we gated our KLRG1 high cells and then looked at TBET and CX3CR1, which are both markers of terminal effector cells, we see a small but consistent increase in terminal effector differentiation. Conversely, when we look at CD127 high cells, which are memory cells, we see a reduction in the mutant cells. And we went on to sort of orthogonally test this using the single-cell RNA sequencing. So we basically took those cells that we had from that last experiment and put them into the 10x, and we were able to essentially test whether the mutant versus the wild-type cells were preferentially occupying CD8 compartments. And so from our single-cell RNA sequencing, we got around 8 clusters. But the main clusters I want you to pay attention to here are cluster 3, which is really the terminal effector cluster, and cluster 0, which is the central memory cluster. And here we've just plotted a bunch of genes that are associated with both of these cell subsets. And you can see that 3 is really expressing the most terminal effector genes, and 0 is expressing the most central memory associated genes. And using cellular density plots, Hannah de Berg in our bioinformatics core essentially found that there was an enrichment of BOC2 mutant cells, specifically in the terminal effector cluster, and a depletion of those cells in the central memory cluster. So this really confirmed to us that that the mutant cells were really becoming more terminal effector-like. However, when we do differential expression analysis between genotypes in each of these clusters, we don't actually see very many differentially expressed genes. But going back to all of our EQTL data, we know that the effect of the variant is likely to be happening in naive T cells, not in already differentiated T cells. So we went back and we looked at our naive T cells from the mutant versus the wild type, and we do see a bunch of differentially expressed genes. And in fact, these genes are very involved with T cell stemness. like LEF1 and MIB, T cell proliferation, and P10 and itch, which are indicative of T cell signaling. So we are getting hints that the effector program may be conferred preferentially in the naive state. And to sort of bolster this data, a study that was published recently looking at stem-like memory cells and knocking out BOC2 using BOC2 guide RNA versus control, we looked to see whether our differentially expressed genes between our mutant and wild-type cells actually enriched for this gene set, and we found that there was quite a substantial enrichment for our BOC2 18-del cells for this guide RNA gene set, with some of the same differentially expressed genes as well. So, oh, this didn't turn out right. But so in other data, we've also made a mouse that is that has the point mutation. So we've basically just inserted the risk allele into the mouse, and we've performed some attack sequencing to see whether we get the same differences in allele-specific reads from attack-seq, and we in fact do. We see these a significant reduction in, oops, this is the risk allele, not the non-risk allele. But anyway, what I'm trying to say is that when we insert this allele specifically into mice, we actually see a reduction in chromatin accessibility, which is exactly what we see in the human. So we hope that we're going to be implementing these models into autoimmune phenotypes and seeing whether there's actually a increased risk conferred. So I hope that I have conveyed to you today that not all genomic assays are equal in their ability to identify causal variants, non-coding variants. The ones that seem to do a good job are ATAC-seq, DHS, and MPRA and CRISPR-I. They seem to enrich most for causal variants in the genome. And following up on prioritized variants using these methods is necessary using CRISPR perturbations. And mouse models can be used to basically understand the in vivo effects of these mutations in disease-relevant cell types. And there's a lot of ongoing research going on with this mouse. We're trying to see if the variant mouse actually recapitulates the deletion findings. We're trying to understand whether there's an effect in CD4 T cells, since there could be an effect on regulatory T cells, and there could be an effect on effector T cells, or there could be both. And it would be really interesting to know whether this specific variant has an effect in all those populations. Does the mutation confer a difference in escape of thymic selection for T cells? And is that a major mode of its effect on these T cells? Do these mutations cause a more rapid onset of autoimmune disease, which is a key experiment that we plan in the future? And furthermore, it's not too insane to think about its role in infectious disease and cancer. But I will say that MPRA was only capable of finding putative causal variants in a third of the loci that we tested. And there's a variety of reasons that this could be the case. First of all, the rest of these loci could just not have variants in regulatory regions that are causing differences in enhancer activity. It could also be because they're acting in other cell types, not in T cells. So we're going to be following up on our MPRA, first by applying it to primary naive T cells. So I told you I did it in jerkats, but we're now doing work to apply these assays in primary T cells, because we think that that's going to be more relevant to human biology. And we also are going to be applying these assays to B cells, specifically GM12878s, because they're very highly transfectable, and we've done the TNFA-IP3 MPRA in these cells with high fidelity. So that's the current ongoing work, and I'd like to thank all of these awesome collaborators for helping complete this awesome project. In particular, Brian Tui and Kosuke, who made the Bok2 18-del mouse. My former postdoc advisor, Nir Hakohen. Michelle Lissner and Ingrid Harden for performing the VSV-OVA experiments. Hannah for analyzing the single-cell RNA-seq data. Michael for performing a lot of the analysis for the MPRA, and Carl for coming up with a very clever way of analyzing the base editing experiment. And I'll take any questions.
So people can either speak up or put questions in the chat. I don't see any questions at this point. People are just rapidly thinking of something. You've got a fair amount of work to do. So you've got like thousands of variants. How are you going to prioritize which mice to make? And you know, I mean, that's always the challenge. Is what I've had to think about.
Yeah.
How do you prioritize what's interesting and what's not?
Yeah. So I think I actually have a hidden slide here. So you set me up for that.
Oh, excellent. Good luck.
So there are not very many highly conserved variants between human and mouse. So so these are the ones that I've mapped anyway. And on the y-axis or the x-axis, sorry, the y-axis here is the probability for causality. So you can see that really above 10% is what I would go after. And there's a bunch of interesting loci where these variants are actually in. Some of these are coding variants, but you know, this limits our ability to create mice from every single non-coding variant that we're interested in.
Well, that's maybe good news. Although I'd remind you that you focus specifically on T cells there, so you're missing, you know, regions of maybe where the mutation is open. The other question I had was, and I looked up that one of the websites that you focused on was sort of the database of immune cell expression. Because you were talking about Bach2, obviously has a lot to do with many cell types, T cells, but also B cells, etc.
Right.
But the region that you knocked out is not open in B cells, so it makes sense that you'd focus on a T cell phenotype.
Exactly.
I think my question is, if I look at this database of immune cells, they've analyzed a lot of immune cells. They've analyzed naive T cells, Th1, Th2, Th17, Tregs, effector, CD4, CD8s, and then they just go to naive B cells. So how do you know that that this area is not open in a cell that's post-activation? Like, it seems like in B cells or any other lineage.
It's a very reasonable question. I've also done an analysis looking at the roadmap epigenetic database, and it seems to be very highly specific to T cells where there's active chromatin. Okay. So we've also done differential expression analysis in B cells, and we found no differentially expressed genes in the mouse.
In the mouse.
But it's still, yeah, you're right. We can't rule it out because we haven't looked at germinal center B cells or you know, other types of B cells. So, but we think because the regulatory regions, you know, we have a good sense that it is very specific to T cells. Yeah. But yeah, you're right. We could probably do more work with regard to that.
I think you've got enough to do. Any other questions? Everyone's been quiet and I didn't see anything in the chat.
Hey, John, when you were looking at kind of selecting out SNPs and doing your screen, is the prevalence of some of these non-coding regions, was the screen done in kind of the European-based population frequencies? Yes. Or
that's a great question.
Have you compared it to the African and Asian populations and looked for selection?
I have not tested variants that are specific to African and other populations. That unfortunately, most high-powered GWAS is done in Europeans, and that it is unfortunate. But I hope that one day we're able to like get many more regions associated with other populations, and we can repeat our experiment in that context. But yeah, we've focused on Europeans for now, mostly because most SNP data, open targets, et cetera, is all almost exclusively European.
But even if you look at allele frequencies, just taking broad databases like, Nomad, do you find any of your interesting targets have population-specific allele expressions?
That's a great question. I have not looked at that, but that's something that I probably should look at.
I had a question, John. John, great work. Really, congratulations. I don't think anyone's ever made a non-coding SNP variant animal model to date. Is that accurate?
There's been one other, I believe. Yep.
So that's super cool, and it's super cool about how you got to the data, and it's really clear. I guess I was curious, like, these things are not selected for autoimmune disease, as you would imagine. So what do you think the protective benefit is? And in what setting could you study it? I know you put infection in cancer. I mean, I doubt that cancer is genetically or evolutionary selecting either, right? It's certainly infection, right?
Yeah. Yeah, I think I was just curious whether there would be a, like, maybe not a selection effect, but maybe a unintentional effect of
protection.
Right. Yeah. Exactly. Yeah. And it could be selective, over generations, for that reason too. I know that in African populations there is, they get much worse lupus. I'm sure that there's some tug of war between infectious disease and autoimmunity in populations that are more affected by infectious disease. It's possible.
Yeah, it's one of the hardest things to test, I guess, with these variants, is to come up with a model where you can, like, we tried this with PTPN22 for TB and couldn't get anywhere.
Right. Yeah, it's low on my list for for nailing down. I think we're going to try to do the autoimmunity stuff first.
Yeah.
Yeah.
I think the parallel studies with the B-cell LCLs would be super interesting to us, obviously.
Yeah, we've already done the MPRA. We just need to the cells are in the freezer. We just need to sequence it and analyze the data. And then, I'd love to be able to get together with some of you and talk about following up some of these variants.
Any other questions? I think that was great. I think you've overwhelmed a lot of people, but it's really, really cool to be able to do this. And yeah, let us know if you want to collaborate on any of the autoimmune stuff, because this is very similar to, as I'm sure you've known, meeting with people, that the types of mice that we've made to try and study things in different cell lineages and induce autoimmunity.
Yeah, absolutely. I mean, yeah, Rich James and I have already talked about potentially modifying some B cells down the line. So it'd be great to get more of you involved in that kind of project.
Excellent. Well, thanks for the talk. It was great.
Great.
And we've been talking to Jane about trying to link that to follow-up of other stuff we've done. So that'd be fun to talk about as well.
That would be great.
Yeah. All right. Sorry we couldn't have you in person today.
Oh, well, thank you so much for having me. I appreciate it.
All right.
Great. Thank you,
Cheers.
John.